All right. Let me start saying that it's a pleasure to be here, and thank you all for coming. Just bear a little bit here with us. It's the first time, I guess we, or at least I, do this presentation through teleconference. So it's exciting. And I'm excited, very excited today to talk about the work that I've been doing the last 1 or 2 years, the work that we have been doing at the Stanley Center as part of a Sweden collaboration group. And let me tell you just a little bit who am I. I'm a computational biologist that has been working at the Stanley Center, part of the Broad Institute, for about 4 years. And I'm going to talk today about rare variants in patients with schizophrenia, in particular patients with schizophrenia collected from Sweden. And so let's start with the very basic background. Why are we interested about rare variants in schizophrenia? Well, the reasoning being that, as most of you know, the schizophrenia is a fairly rare disease. Less than 1% of the population is affected. At the same time, irritability is it's very strong, from as has been ascertained from clinical study. So most of the variability for the schizophrenia phenotype is believed to be due to genetics, and despite the fact that the genetics is it's so important, we know from studies that people that are affected with schizophrenia, and this is true also for other psychiatric diseases, have a severe effect on fecundity, which means that they have far fewer offspring on average than the people that are not affected with the psychiatric disease. So, for example, if we take yes, I guess yes, you can see my mouse, which is nice if you can, so males with schizophrenia on average have less than half than the expected number of kids of the average person. So the idea here is that clearly if there is a variant that has a very high penetrance, or maybe even full penetrance for the disease, it's clearly is not going to last in the generation for a long time, because it will be selected out of the population just by this simple observation. Therefore, if we want to find variants for schizophrenia and other psychiatric diseases with strong effect on fecundity, if you want to find rare variants with strong effects, we really expect to find these only in the rare part of the allele frequency spectrum. And we have seen some examples of rare variants in psychiatric diseases in the past. We have seen examples in the work done with respect to CNVs. So it is even before we were able to do next generation sequencing experiments, so analyze the sequence of people at many regions in the genome at the same time. And the CNV literature has taught us maybe a story that we would kind of expect from what I said before, that even if CNVs are not fully penetrated, there are CNVs in schizophrenia that have very strong penetrance, very high odds ratio, and we don't see this as segregating the population, but we see this as arising, maybe not arising in the last generation, but we see this as arising in the last few generations, so that each CNV that we see in a patient is kind of an independent event from CNVs that we see in other patients. And again, this is reinforcing the idea that yes, these are events that happen and likely to be selected out of the population because they have such a strong effect on fecundity. And the reason also that we were able to see CNVs, and now the literature on CNVs maybe is a decade old, is also that CNVs are kind of recurrent events. in some cases, and so even if they're independent events, sometimes they happen in the same regions, and then we're able to build statistics for this and to associate them with the disease. Now, for non-CNV mutations, the statistics might be a little bit harder, but nevertheless, for the purpose of this talk, we're gonna look at rare variants in the exome of about 12,000 within individuals that are more or less divided half and half and in controls, like so people without psychiatric phenotype and people with schizophrenia, so schizophrenia cases. And on top of that, we're going to inform our analysis using a cohort of 45,000 individuals. Maybe some of you have heard before, this is the EXA cohort. It has been collected as a joint effort led by Daniel MacArthur. And we're going to inform the variants we find in 12,000 individuals using this even larger cohort of 45,000 individuals. And the reason we do that is that, again, we want to focus our attention on ultra-rare variants. So variants that are as shifted as possible in the allele frequency spectrum towards things that are rare. And again, we do that because we want to concentrate our attention on things that have the highest likelihood to be recent and have arisen in the last few generations, if not even maybe in the last generation, so as the novo variants. Again, parenthesis, because this cohort was collected as a population cohort, we don't have DNA of parents, so we are not able to distinguish the novo variants from inherited variants, which would be nice, but just for the logistic of how these studies was designed, we cannot do that. But anyway, we focus on ultra-rare variants, and just to give you an idea, ultra-rare variants will be defined as variants that are observed only once in the whole cohort. So across about 25,000 chromosomes, only one chromosome as that variant. And also there are variants that are never observed in the exact cohort. So something like with an allele frequency is less than 1 in 100,000. And although these things are very rare, every individual across the genome or across the exome, because we only analyze the exome, will have a few of these. And while they account only for a small percentage of the heterozygosity, we still find maybe something like 30, 40 ultra-rare variants in each exome. Now, we are going to focus for the purpose of the presentation mainly on this set of variants. So, but we further divide these into 4 groups. And these groups will be synonymous, missense non-damaging, damaging, and disruptive. So synonymous are going to be variants in the exome that, although they change the DNA, they are predicted to not affect the protein product of that particular exome. Missense variants will be variants that affect the protein product, and we will divide these in 2 groups, missense non-damaging and a small subset of variants that we call missense damaging. Missense damaging will be variants that are classified as damaging by a different set of algorithms, including also inflamed indels and protein-protein contact domain variants. And missense non-damaging are going to be all the other missense damaging that don't fall in the damaging category. And disruptive variants are going to be the ones that are likely to be the most affecting proteins, and you might have heard of them as also as protein truncating variants, and it's everything that you can classify as loss of function or that causes nonsense mediated decay. The idea is disruptive variants will be variants that affect the protein in a very deleterious way. Now, if we look at the association of these variants with schizophrenia, we see already something interesting. So in aggregate, we see that individuals with schizophrenia have an excess of damaging and disruptive variants, but they don't have an excess of synonymous and missense non-damaging variants. And this is also important because it means that likely this results is not confounded by possible biases in the analysis. And you're gonna see a few of these plots where on the left I show the excess of a given set of variants in samples per case, and on the right you're gonna see the odds ratio of that category of variants. And the first thing that we see in this plot is that schizophrenia patients on average have about 0.25 of these either damaging or disruptive ultra-rare variants. And this is on a background of about 4 damaging or disruptive ultra-rare variants, which correspond to something like a 7% enrichment. That means that these kind of variants overall have a 1.07 odds ratio for association with schizophrenia. So not a strong odds ratio if you want, but this is because we're looking at them in aggregate as an average. It's clear that some will be probably very have large effect size and some are probably irrelevant. But nevertheless, it is interesting that we already see this enrichment on an exome-wide level. And so then the next thing is that we can ask is, first of all, like, is this an excess that could be explained by maybe de novo variants? Maybe we see more variants in schizophrenia cases because schizophrenia cases have more de novo variants than controls, and maybe they just accumulated with this excess in the last generations. But interestingly, what we can infer is that it is not possible that this class of variants is de novo. It must be the case that mostly these variants are inherited, and maybe I'll skip the math here a little bit, but if we look at the rate of de novo disruptive and damaging variants in schizophrenia cases and the rate in controls, the delta, the difference between these two rates cannot explain this excess of 0.25 disruptive damaging variants we see in cases. And so these disruptive damaging ultra-rare variants we are going to call them DURVs, which is kind of an interesting term. It sounds pretty good. And I'm going to use it a little bit more in the talk. And again, so we are going to think of these variants as variants that mostly are inherited, but yet we're still thinking of them as variants that most likely have arisen in the last maybe 10 generations. Now, if we look at this kind of variants, those disruptive and damaging across single genes, and maybe we even go a little bit beyond ultra-rare variants, we take things that are a little bit more common. Do we see any gene associating? Well, the answer, unfortunately, is not. Probably because of power, because anyway, we're testing many genes. We don't see any gene that associates to a level that would make it unequivocally associated with schizophrenia. Although this is unfortunate, this makes sense in the view of the fact that we know schizophrenia is a very, very, very polygenic disease. And what this is telling us is that even if we see an enrichment at the exome-wide level, but we don't see an enrichment at the single gene level, it is likely that there is no single gene that is responsible for even 1% of this enrichment. There are probably hundreds of genes that are responsible for the enrichment we see. So even if we cannot identify any single gene in this cohort, and we probably will be able to identify a single gene with larger cohorts, what else can we do? And as many was telling us before, we can ask whether the enrichment concentrates in specific gene sets rather than just asking whether this enrichment that we observe concentrate exome-wide or in single genes. And so the next slides I'm going to show are going to be mainly referring to gene set analysis. So first of all, we ask the question of whether DURV variants, for which we see a burden in schizophrenia cases, can implicate one tissue compared to others. And there are many ways to do this analysis. I'm not going to go into the details, but pretty much any way you define a set of genes that are specific to brain, you'll see that this set of genes is much more enriched than similarly defined gene sets for other tissues like colon, heart, kidney, and others. And this is already per se very interesting, maybe expected, because what we see is that these DURVs really, this excess of DURVs really concentrate in genes that have to do with the brain. And we can push this further. We can go beyond tissue. We can go to cell types. So if we look at cell types in the brain, again, we can see that this enrichment of DURVs concentrates in cell types that have to do with neurons, and not, or at least not as much, in cell types that have in the brain that are not neuron-like. And I'm going to say this is, first of all, it's really cool because it's the first time we were able to do this with rare variants with so much signal. But it's also interesting because it tells a story that is parallel to the story that is being told by the common variants, if you're familiar with that. And okay, then we can try to push this a little bit more. For those of you that are more familiar with the literature on schizophrenia and rare variants, you might know that there are other gene sets that have been shown as very associated, like, for example, FMRP genes. So genes that are target of the FMRP protein have been shown as enriched for rare variants already in schizophrenia. They have been also shown as enriched in autism. And one hypothesis that we had is maybe these genes, they might be a proxy for genes that not only are in the neurons but are in the synapses in particular. So how do these gene sets fare when we analyze them with respect to this DURV excess? Well, we see a strong excess, and in some ways we see an excess that it's even stronger than genes that are specific to neurons. So there does seem to be something about genes that we call potentially synaptic, as they show a stronger enrichment even in the neurons. And in particular, it's interesting to see that if we put some of these potentially synaptic gene sets together, they might explain, they explain at least 70, might explain even all of the enrichment that we observe in this cohort. And for those of you that are familiar with other results, for example, like the ARC gene complex, the NMDAR gene complex, and other like protein complexes of the synapses that have been associated in the past, how do these gene sets fare in this kind of analysis? Well, they do replicate. We do see an enrichment in these complexes. However, and I'm not going to talk too much about this slide that contains also other results, but I do want to tell you that in this analysis, we're able to see that while these gene sets replicate, they only explain a small fraction of everything that is going on at the rare variant level. So not to say that they're not important, they probably are important, but there is also so much other stuff happening, we guess, at the synapses that is being disrupted in schizophrenia patients. And we also see, I'm going to say just very shortly, we also see enrichment in other categories of genes, like intellectual disability genes on the X chromosome, genes that have been unequivocally associated with developmental disorder and developmental delay. And I'm not going to spend too many words there, but that's also an interesting observation that we're able to come up with this cohort. How do these results compare with other results in the literature? So, for example, common variants and CNVs. Well, for those of you that are more familiar with the association of schizophrenia with common variants, if we aggregate common variant signal across the genome, we can build scores that explain something like by 15% of the phenotypic variability in schizophrenia samples. at least in this cohort. CMV explained maybe 0.7% of the variability in schizophrenia. If we take DURVs in aggregate, and again in aggregate, so taking average effect sizes in potentially synaptic genes, we explain maybe something like 0.9% of the phenotypic variability. So maybe little compared to common variants, but this has to be taken as a lower bound, and this is going to increase as we refine the statistics for each variant. And so to conclude, the main messages that I want to make sure that stick is that we are able to see an excess of these DURV variants, so disruptive or damaging variants in ultra-rare variants. So on a background of maybe 4, we see 0.25 more in schizophrenia cases, suggesting that at least 1 in 4 schizophrenia patients carries one of these very ultra-rare and disease-relevant variant. Of course, I remind you, we're only looking at the exomes. This enrichment in DURV, it's very polygenic, so we're not really able to say much about what kind of genes are affected, but it does seem to be compatible with an enrichment of genes that are predominantly expressed at the synapses of the neurons. And this should also tell you that a comprehensive high-quality annotation of synaptic components will definitely benefit further interpretation of the signal as we go with larger sample sizes and we are able to further dissect the signal. With that, these results will be available in the Nature Neuroscience journal in November. And I want to thank the many people that have made this study possible. This, of course, it's the result of many years of work and many PIs that have got together to generate this Sweden cohort. And I'm also excited to say that Andrea Ganna here is also going to show some results from this cohort and talking about ultra-rare variants with respect to cognition rather than schizophrenia. And with that, if there are any questions, I'd be happy